So hello everyone. Thank you so much for joining us, joining our bioinformatics and genomics seminar today. So since Piotr and Shira are not here, so Dr. Ann, Aldana, and I will co-host this seminar. So I'm actually the senior bioinformatics scientist from research computing team. And before Ann actually introduce our speaker, Dr. Andrew Sharp today. So let me just briefly introduce important resource from the research computing team. And we actually record what our seminars in YouTube. So let me share my screen. So if you type RC Wiki in a browser, and you will go to our research computing wiki page. And in this page, we actually record all our seminars in YouTube. So if you scroll down to this page, you will find the digital science education page. So click this link, and you will find all the seminar schedules. Like say, for example, on July 14th, we have a lot of speaker, Michael Wangla, who will speak his genetics in medicine paper. So if you are interested, you are welcome to join us. And if you scroll down, we actually record all our seminars in YouTube, and we have the videos, what are YouTube videos on this page. So if you find any interesting talk, so feel free to watch that later. So I think that's what we have. So Anne, can you introduce our speaker today?
Absolutely.
Thank you.
It's my pleasure to introduce Andrew Sharp, who's an associate professor in the Department of Genetics and Genomic Sciences at Mount Sinai School of Medicine in New York. He has a really diverse training background. I think has influenced his lab's focus today on reverse genetics to apply a bottoms-up approach to understand how genetic and epigenetic variation underlies human disease. So he initially trained in graduate school with Dr. Pat Jacobs, University of Southampton, studied X activation and epigenetics in disease. He then did postdoctoral research with Dr. Evan Eichler at the University of Washington, where he studied copy number variation in human disease. And then to further expand his training, he did a Marie Curie fellowship at the University of Geneva in Switzerland with Dr. Julianos Antonarakis, where he evaluated methylation profiles in disease, including at imprinted loci. He's won a number of awards for his research, which has been published in many high impact journals that many of us have seen already. And today he's going to be telling us about some new work that he has recently shared as a preprint on bioRXiv that I also actually got to hear a sneak preview of at ASHG last year. And personally, it was my favorite talk of the meeting because it's some really exciting work showing the role of under-recognized role of methylation in human disease. So thank you, Andrew, for joining us today to tell us about your work on a survey of rare epigenetic variation in 23,116 human genomes to identify disease-relevant epivariations and novel CGG expansions.
All right, thank you very much for that nice intro. I'm going to share my screen. Hopefully you guys can all see that. Thank you very much for inviting me. I guess in normal times I might be with you in Boston right now, but here we are sitting at home or in our offices. So I'm going to talk about some work that I guess spans in total about the last 5 years we've been doing in my lab. Most of what I'm going to talk about today is this more recent study we've been doing in the last year or so that, as Anne said, I presented at ASHG last year. On my first slide here, I put some references. So we posted this on bioRXiv a few months ago, and probably tomorrow I'll be submitting the revised manuscript to American Journal. So hopefully this will come out in the next few months. If any of you are interested after seeing this talk, I'm just going to very briefly touch upon 2 other papers that we've published on this same theme. The first paper here in Nature Communications, where we looked at patients with congenital disorders, specifically looking for epigenetic variations that were linked with disease. And then also a follow-up paper where we looked at a cohort of about 1,000 patients with autism and schizophrenia. Okay, so if we think back through some of the history of modern genetics in the last 2 decades, there's been some really important basic studies like the HapMap project, the 1000 Genomes project, and then aggregation consortia, things like the exome aggregation consortia, that have given us huge leaps in our understanding of both common and rare variation. So for example, the HapMap was what established large-scale maps of common variants that facilitated genome-wide association studies. Things like the exome aggregation consortium have given us insight into rare variants, and these have been really fundamental in things like exome sequencing studies, which have really been, I would say, the drivers of most modern insights into human genetic disease. But it's important to realize that DNA sequence variation isn't the only kind of variation that can modify phenotypes. So what I'm showing you here is really, I'd say, the sort of groundbreaking study in this area. And this was published just over 20 years ago in this mouse model called the agouti mouse. And what they showed here is that altered methylation of a transposable element upstream of the agouti locus is what drives phenotypic variation in this mouse. And you can see this very clearly based on variation in coat color, but also it actually caused variation in a variety of metabolic defects as well. And if you then dig into the human genetics literature, over the last, I'd say 15 or 20 years, you'll actually find there's quite a number of studies that have identified epigenetic defects that contribute to human Mendelian disease. So for example, here is one paper where they took a cohort of patients with hereditary colon cancer and found that about 2% of these, the hereditary colon cancer, is not due to point mutations, but in fact appears to be caused by promoter methylation of the MLH1 gene, one of the main drivers of hereditary colon cancer. Here's another paper that appeared more recently. Here, this paper described a couple of families where there was promoter methylation of BRCA1 in families with breast ovarian cancer. And another paper here that looked at a particular type of metabolic defect. And this found that this defect of vitamin B12 metabolism was sometimes caused by promoter methylation of the MMACHC gene. And these, unlike the previous two, was a recessive disease. Sometimes these epimutations were homozygous, sometimes they occur as compound heterozygous with point mutations in the same gene. So there's really quite a lot of evidence out there to say that epigenetic changes can be important drivers of human phenotypes. But really, in contrast to all this data that we have on sequence variation, there's no large-scale population surveys that have been done to assess the prevalence of this kind of rare epigenetic variation in the human population. So just before I keep going, I just wanted to introduce this concept of different types of epivariations. So originally when what I'm referring to as epivariations now were described, they're often called epimutations. And in this form, people thought they were generally purely epigenetic defects. And they were, in humans at least, first described in association with, typically with imprinting errors. So for example, there have been epigenetic errors associated with Angelman syndrome, where there are no sequence mutations that people can identify, but instead there just appears to be an imprinting defect so that the imprinting mark is not properly established, resulting in Angelman syndrome. So primary epivariations are thought to be just purely epigenetic, spontaneous or sporadic errors where for some reason the cell or the embryo doesn't set up the methylation mark in the correct way. In contrast, other types of epivariation we can refer to as secondary epivariations, and these are attributable to an underlying mutation of the DNA sequence. So probably the best known example of this would be Fragile X syndrome. We now know this is caused by an underlying CGG triplet repeat expansion. And when you have a large expansion of more than 200 copies of a CGG repeat of the promoter of FMR1, This causes FMR1 to get highly methylated, switches the gene off, and then in males with a single X chromosome, they're now nullosomic and suffer from Fragile X. And just as an interesting sidebar, Fragile X is termed Fragile X because way back in the days of cytogenetics, this disease was actually diagnosed by, you would culture cells from a patient you suspected that might have this disorder in a medium lacking folic acid. What you would see is that the tip of the X chromosome would actually break off in a proportion of cells, which is called a fragile site, hence the name of Fragile X. It was subsequently described that in fact this fragile site is actually the CGG expansion itself, and this is thought to be due to the fact that these CGG expansions have very strong secondary structures which polymerases have a great difficulty traversing, causing these double-strand breaks during DNA replication. I'll come back to this a little bit later. So really, the aims of our study were pretty basic. That is, how prevalent are epivariations in the human genome? And then we wanted to understand, what are they doing? What is their functional impact? How might these things contribute to human disease? And then also we wanted to try and understand what are the possible causes of epivariations? How many of them are primary? How many of them are secondary? And then other questions such as, are these potentially heritable between generations? And then finally, something I'd sort of suspected for a while based on this idea of Fragile X is that what's the contribution of repeat expansions as a cause of epivariations? And I'll touch on all of these different topics as I go through the talk. And so to start this study off, I really went about just trawling through databases that are out there to try and see how much DNA methylation data I could scrape together. And I ended up focusing on 22 different cohorts, each of which had used the Illumina 450K array to profile DNA methylation across the genome. And I specifically chose basically any cohort I could find that was on GEO, dbGaP, Express, EGA, and even some sort of private databases where they had at least 300 individuals profiled with the 450K array. All these cohorts are listed here. Most of these are just simply population cohorts. A few of them are individuals with specific diseases such as facial clefts, rheumatoid arthritis, or Parkinson's disease. Most of them are just general individuals from the population. Importantly, 4 of these cohorts include samples of twins, either monozygous or dizygous twins. 4 of them have gene expression data available, and also 2 of them had SNP array data available. And we use this as secondary data sets to give us some additional insight into the causes and consequences of these epivariations. And so in total here, we had just under 25,000 individuals, each of which had data on nearly 500,000 CPGs. Just to point out, the Illumina 450K array is not fully genome-wide methylation. It's looking at a targeted set, like I say, of about 500,000 CPGs, specifically focused on gene promoters, CPG islands, and a few regulatory elements. And we had more than 700 pairs of monozygous twins, nearly 8,000 individuals with expression data, and a few thousand individuals with SNP array data. And just to point out, all of this data I got for free from the internet. A lot of it involved, filling out data access agreements, things like that, IRBs. But during this whole study, we actually did very little data generation ourselves. And so I think there's an important lesson there for people out there. You don't need a lab. These days, there is so much data available. You can really just take things that other people have published with other questions in mind. And if you have an interesting idea, an interesting hypothesis, like I think we did here, you can actually do some very well-powered studies simply by taking other people's data and repurposing it for a new question. Now, the problem of taking other people's data is sometimes it's crap, I will say. So a really important step in our study was doing some pretty intensive QC and data filtering, because there were actually a few more studies I didn't list here that we just threw out completely because the data quality was very questionable in some cases. Basic steps we did. Each individual, we calculate the fraction of probes that have a low signal intensity, and where that's more than 1% of all probes on the array, we throw that individual out. So these are typically marking poor hybridizations, or where there was insufficient DNA being hybridized, things like that. If you don't do that, what you find is we end up calling in some individuals even hundreds of outline methylation loci that are nearly all false positives, just because the data is not very good quality. We also perform principal component analysis. Again, this is another very important way of getting insight into these large data sets. And so here I'm just showing you an example plot. What you're looking to see is hopefully a nice tight cluster of the samples, but where you see people that are outliers, we basically throw those out. It's a qualitative step, but it's a very good way of just recognizing outlier data points. You don't usually know exactly why they're outliers, but we took a conservative approach and threw them out. Another very important step that we did here is we calculated the proportion of blood cells that were contributed by each of the major types constituent in whole blood. This is very important because most tissues, blood is one of them, are heterogeneous mixtures of multiple different cell types. And here is because we're looking at epigenetics. Epigenetic profiles differ between different cell types. And so what we were trying to avoid is having lots of false positives that maybe, for example, if you had somebody with leukemia who has a huge spike in their B cell content, for example, if you have genes that are specifically switched on or switched off in B cells, but on other cell types, they're going to show up as epigenetic differences that are not due to anything other than the fact that there is a variation in the underlying fraction of B cells in their blood. And so we've removed outliers for blood cell composition. And they also did a very simple check over here on the right, looking at predicted sex from the data versus reported sex of each sample. So the plot here I'm showing you on the x-axis, we take the mean beta value on the X chromosome. The y-axis is the fraction of probes that failed detection p-value on the Y chromosome. And so you see generally two very nice clusters, males down here in the bottom left that have a single X, a single Y. Females up here in the top right that lack a Y chromosome but have two X's, one of which is highly methylated. And so this spots half of your sample mixes. So some samples labeled as male will show up as female and vice versa. Usually that's because a sample got switched somewhere along the line. We threw those out. It will also spot individuals with sex chromosome aneuploidies. So this is probably a person with Turner syndrome, a single X and no Y. These are probably people with Klinefelters, so two X's and a Y. To be conservative, we also discarded those. And so after all these QC steps, we actually threw out nearly 2,000 individuals, about 7.5% of our data, and ended up here with this number of just over 23,000 individuals we use for downstream processing. Again, another very important step here is we normalized our data. So we used a pipeline using the packages Lumi and BMIC, which do a number of different steps: color correction, dye bias, intensity, and then normalizing for the fact that you have two different probe types on these arrays. And then we filtered at the probe level. So we removed probes that overlap common variations because they can give you some false positives. We removed non-unique mapping probes and those with low signal intensity. And then to actually identify regions of outline methylation, we used a sliding window approach, which is quite simple. The precise definitions are here, but basically what this does is we take a 1 kb sliding window, we slide that through the genome, and we're looking for regions in the genome where you have at least 3 contiguous probes, all of which lie within the 0.5th percentile of the distribution and have a difference of at least beta value of 0.15 above or below the population mean. That might sound complicated, but if I show you some plots, it'll make perfect sense. So this is the kind of thing we're looking for. This is a rare gain of methylation that we see in one individual here shown in red. At the bottom of the plot here are the other 23,115 samples. Just to orientate you, the x-axis is genomic position. So we're looking at a region of about 1.2 kb here. The y-axis is the methylation level. So 0 is unmethylated, 1 is fully methylated, 0.5 is consistent with one of your two alleles being methylated. We don't get allelic data from these arrays, but you can kind of infer that from the data. And so, as I said, the 23,000 individuals are shown here. Each one is individually plotted. So these dotted lines, each one is the methylation value in one individual. But over the top, we put the population average as the solid black line, and these shades of gray are 1, 2, and 3 standard deviations from the mean. And so here is an individual with an obvious outlier methylation called by a sliding window. Other kinds of changes we see are things like this, where we have recurrent gain and loss at a single locus. Here we have, about 10 or 15 individuals with gains, 10 or 15 individuals with losses. So just to summarize, give you some sort of headline numbers from these 23,000 methylomes. In total, we found just under 14,000 epivariations in about 7,500 individuals. That is, at the criteria I specified, about 1/3 of our cohort has one or more epivariations identified in their genomes. There's nothing magic about the criteria we used. It's pretty stringent. Like I say, the majority of individuals we call 0 methylations, epivariations in their genome. But as I'll show you with some validations, nearly everything we're calling is correct. So it's always that trade-off of false positive, false negative. We set it a fairly high bar, but the things we're calling we're pretty sure are real and genuine large outlier events. These nearly 14,000 epivariation calls occurred at about 4,500 unique loci in the genome. We see about a 2-fold excess of gains to losses. That may largely be driven by the fact that the array we're using is targeting gene promoters, which inherently tend to be unmethylated. And so obviously at an unmethylated locus in the population, you can only ever see gains. Important to note, this frequency plot over here on the right is showing that the majority of epivariations we see are singleton events. That is, in all 23,000 people, we only observe one individual that carries that. And then as we get more and more common, we see less and less of those in the genome. And we saw a small number, about 28 loci, that show both gain and loss at the same location in different individuals. So we just wanted to be sure that things we're calling by our array approach are real. So we took a few loci, obtained the DNA sample from the individual that was called with an epivariation from the array. Here I'm showing you the profiles on the array. Whoops, I just lost my share screen. Let me see if I can get that back. I have. Okay, hopefully you can all see that again. Sorry, I don't know what happened on my computer. And here I'm showing you where we performed bisulfite PCR and high throughput sequencing of each of these 4 loci. On the left is the person with the epivariation, on the right is the control. And in each of these 4 loci, you can see that we see a population of highly methylated reads that is absent in the control, basically replicating the array results with this secondary methodology. And we've actually, in prior studies, done this on about 60 loci, and we get a true positive rate of in excess of 95%. So we then wanted to see what are the effects of these epivariations on genome function. And so here we looked at gene expression, and we used data from the BIOS cohort where we had RNA-seq in about 3,500 individuals. And if we focus specifically on places in the genome where we see losses, methylation at gene promoters, what we see here is that these individuals with promoter hypomethylation tend to have unusually high expression levels in the population. So the y-axis here is the rank of the epivariation carrier in all 3,500 individuals. And you can see many of them actually have the highest expression in the whole population. If we then look at genes that have promoter hypermethylation, that is gains of methylation of the promoter. We see the opposite, where we see most of these individuals have unusually low expression levels. And this is entirely consistent with what we know about gene promoter methylation, that is generally a repressive event. And this was highly significant by permutation analysis. Just to firm this up a bit more, we then used some data from the 1000 Genomes project. We also called epivariation those. Here, the nice thing was there was allelic expression data available. And so, for example, here we have an epivariation called at the UB2T gene. Just to note, this is a gene where if you have biallelic hits, you get a Fanconi anemia. So this is one where we predict this might be linked with human disease. When we look at the carrier of this epimutation, we see they actually have the lowest expression in the entire 1000 Genomes cohort. And then when we look at the allelic expression data, here we have a transcribed SNP, a C to a T, which I'm plotting just on this scatter plot. Everybody else that has this C to T variant has nice biallelic expression, whereas our epivariation carrier here has about 80 reads showing C and 0 reads showing T, so they have monoallelic expression. So therefore, the array data is picking up a promoter hypermethylation event that's associated with unusually low expression, and this is because one of their two alleles is switched off. So this would be basically functionally equivalent to having a stop codon in this gene. Just to add a little bit more, we also looked at what epivariations are doing if they're not located at gene promoters. So over here on the left, this is epivariations that are within a gene body. So somewhere within the start to stop of a gene. Here we see a positive association. So if you have gain of methylation, you have slightly higher expression, loss of methylation, slightly lower expression. It's much weaker than we see at promoters, but it's significant. When we take epivariations that are not overlapping a gene, i.e. an intergenic space, and we relate it to closest gene, we didn't see any significant association there. But we have various issues. There are less of these, we have less power. We don't know what direction these may be. Some may be repressive, some may be enhancing. And we don't even know if the closest gene is the relevant one. So I wouldn't read too much into that. Basically, it's a harder problem to solve. And we then ask the question, what's the relationship between promoter epivariations and selective constraint of genes? So here we simply took PLI scores generated by the Exome Aggregation Consortia. PLI stands for probability of loss of function intolerant. So the higher the number, the more constrained that gene is. The violin here on the left is all the genes that we tested on the array. And when we take genes that had gains of methylation of the promoter, we see they have unusually low PLI scores. And the same is true for genes with losses of methylation, but to a lesser extent. So basically this is saying genes that are hit by epivariations tend to be less constrained, which is consistent with purifying selection operating on promoter epivariations. And if we then look at that in relation to how common are epivariations, so rare ones that we only saw in one person on the left, more common ones seen in at least 10 individuals, we see that PLI scores go down as these epivariations become more common. So again, consistent with selective constraint operating on many epivariations. And this is all pointing towards the fact that these are likely doing functional things that are acted upon by natural selection. So based on this expression and constraint data, we then took our epivariations and overlapped these with known disease genes from the OMIM catalog. And we can find more than 400 OMIM disease genes that have promoter gains of methylation, many of which we would predict result in silencing of those genes. So obviously I can't list all of those here, but just a few highlights. So we hit, for example, MMACHC, which had previously been described to cause metabolic defects. We found that at a frequency of about 1 in 1,000 individuals in the population. Another gene here, XYLT1, is found to have promoter methylation caused by underlying CGG expansion. We found that segregating at about 1 in 2,300 individuals in the population. But obviously we have hundreds of other genes which we would predict epivariations of these likely form part of the mutational spectra underlying numerous different Mendelian diseases. And so my guess would be, if you're somebody that has your favorite disease and you have patients where you can't find a coding mutation, if it's in our catalog, you might want to start looking at promoter methylation as a possible cause of some of those unexplained patients. Just to hammer this point home, the American College of Medical Genetics has published a list of 59 genes that they consider to be pathogenic and medically actionable. That if you find point mutations in these genes in an exomal genome sequencing study, you should tell the person about them, because that's something important they should know. These genes are listed here with the estimated prevalence of these epivariations on the right. Just to add some color to that, BRCA1, the most common cause of hereditary breast and ovarian cancer, we found 7 patients with gains of methylation of the promoter. And another one, LDLR, the low density lipoprotein receptor. This is associated with hypercholesterolemia. So that gives you a very high incidence of coronary artery disease. We found 4 people with what we would predict would be inactivating promoter epivariations, this gene. Now, gains of methylation of the BRCA1 promoter, as I already said, have been reported in the literature before, but this was reported in the context of, presumably these are very rare events. They just reported 2 families. When you actually look at our data, we found 7 carriers, which gives us a population incidence of around 1 in 3,000, 1 in 4,000. If you compare that to the incidence of BRCA1 coding mutations, which is what everyone looks at today, that's around 1 in 200, 1 in 300, depending on what cohort you look at. And so if you do the math, that suggests that perhaps in the order of 5% of pathogenic BRCA1 alleles are in fact probably due to promoter epivariations. So that again suggests this is something people should probably be looking at. So now I'll just switch gears slightly and talk about some of the downstream things that we did. So we now wanted to get some insight into, sorry, my slides have, something keeps happening with my computer. Okay, I'm back again. We wanted to get some insight into these secondary epivariations, get some insight into the causes of epivariations and what is causing these things. And so the first thing we did is we took a cohort of nearly 1,000 individuals where we had SNP array data available. And we took each epivariation that we found in at least 2 or more individuals and used that as the substrate for an association analysis. So just like a genome-wide association study, where you're associating a phenotype with a variant. Here our phenotype was, do you have the epivariation, yes or no, and your SNP data. And we looked plus or minus 1 megabase around each epivariation, and the plot here I'm showing you is the kind of thing we observed. So this plot is showing plus or minus 500 kb, plus or minus 1 megabase around the epivariation, which is in the middle, indicated by the red line. We have minus log 10 p-value, so this is a Manhattan plot. 1% FDR threshold is shown by the red line. And what you can see is that we have this very strong signal of association that actually overlaps the epivariation. Our peak SNV here, this has a minor allele frequency of about 1% in the normal population. But here we had 9 carriers of this epivariation, and 8 of the 9 were heterozygous for the SNP. That gives us a very strong p-value, about 10 to the minus 40, and tells us that this particular epivariation is clearly a secondary event that's segregating on a local haplotype marked by these particular SNPs. And overall, out of the epivariations we're able to test, 70% of them had some signal of genetic association at 1% FDR. So this is telling us that the majority of epivariations are likely to be secondary events, segregating on or being caused by some underlying genetic change. Just to give you a few more examples, here I'm showing you 9 more plots of these association that we get. The top 6 here, what we typically observed is that the region of association was physically overlapping, or for example here, very, very close to the epivariation. So telling us that there is some genetic variant either within or very close to the epivariation itself that's causing that. But at the bottom, here's 3 examples of epivariants where in some cases we have association signals more than 500 kb away. So in some cases there can be quite distant epigenetic variation, sequence variation that's causing an epigenetic change somewhere else on the chromosome. Now we then dug into this in more detail. So we actually then looked at a different cohort, the PPMI cohort, where we had just under 500 individuals where we had methylation data and whole genome sequence. And first we looked at rare SNVs and associated these with the presence or absence of an epivariation. And what we found here is that if we focused on very rare or private SNPs, the plot here I'm showing you is the relative density of rare variants in epivariation carriers shown by the red line and controls in the blue line. And you can clearly see that here we're looking plus or minus 5 kb around each epivariation. There's a very strong enrichment right on top of the epivariation itself for the presence of rare SNPs in the people that have these epivariation events. This is a highly significant P of 10 to the minus 63 and nearly 11-fold enrichment. If we then take SNPs that specifically overlap transcription factor binding sites, this is based on ChIP-seq data from the ENCODE project, even though the numbers drop, our enrichment and our p-value increase substantially, and you can see the region of enrichment is right on top of the epivariations themselves, just within a couple hundred base pairs of the epivariation midpoint. This is telling us that a fraction, we estimate to be around 8% of these epivariants are specifically caused by rare variants that are disrupting transcription factor binding sites at the epivariation itself. Just to give you the underlying numbers, out of the 370 epivariations we could test, 33 of those had a rare SNP at them, which was about a 10-fold enrichment versus background. So we estimate around 8% of epivariations are due to these rare underlying SNPs, many of which are disrupting known transcription factor binding sites. We then went on to look at copy number variants in the same cohort. Here we looked CNVs are at least 2 kb in size within plus or minus 50 kb. And again, we see an enrichment. Just to show you one example, here's an individual that has a 36 kb duplication. You can very clearly see that. So the y-axis is the estimated copy number from CNV data, and you can see it goes up to 3 copies here. The other 500 individuals have no CNVs in this region. When we look across all of the epivariations, we had these rare CNVs at 10 of them. So it's a pretty small fraction, only about 2 or 3% of our cohort. But again, this was highly enriched versus background, a nearly 40-fold enrichment with a p-value of 10 to the minus 71. So again, this is telling us a small fraction of epivariations, a few percent, are caused by rare underlying CNVs. Now, we then harnessed one of the other aspects of our cohort, which is the fact that we had monozygous twins. So we had 700 pairs of MZ twins. And what we wanted to do is ask, because MZ twins are derived from a single progenitor embryo that then splits, you would expect that they must have the same genetic sequence. And we basically looked at concordance in these twins. So if you look across our MZ twin pairs, this is what we observed, 63% of these epivariations, that both twins have the same epivariation. When one twin has it, so does the other, which is what you might expect. But surprisingly, we found that actually 30% of the epivariations that we saw in monozygous twins, one twin carried it, but the other one had a completely normal epigenetic profile. And then a small fraction, one twin was a clear outlier, the other one was a minor outlier to a lesser extent. And so overall, this is telling us that around a third or so of these epivariations have a somatic, that is a post-zygotic origin, which was potentially consistent with somatic mosaicism. I'll actually just skip this slide because I see time is running on. We then went on to look at inheritance. So we had 117 parent offspring families. And we wanted to ask the question, if an epivariation is found in a parent, how often is it passed on to the children? So we had 40 epivariations found in parental samples, giving us a total of 89 informative meioses. 31% of these epivariations, or these informative meiosis, here we can see a plot where the mother in red has an epivariation that's passed on to one of her 3 children, whereas other loci look like this. This was 69%, where a parent has an outlier methylation mark that none of their kids inherit. And overall, this is significant departure from the Mendelian expectation of 50%, suggesting or showing us that epivariations are less heritable than you would expect under normal Mendelian rules of inheritance. And this is consistent with some of them either being somatic mosaics, so these events are absent from the germline and therefore not passed on, or potentially that some of these may be purely primary events that are present in an individual, but then are wiped by epigenetic reprogramming that occurs during gametogenesis and what goes on in the early embryo. Just to emphasize that a little more, we then looked at the relationship of epivariations with age and found that as individuals became older, you tend to accumulate more epivariations. So people with more epivariations are statistically older than people that have few. Again, highly significant. And we also, when we went back to our monozygous twins, found that twins who are discordant for epivariations tend to be older than twins that are concordant for epivariations. So again, this is telling us some of these are somatically acquired with age. As a counterpoint to that, if you then take individuals where you have multiple different tissues, what we generally observed is that if you see an epivariation present in one tissue, you'll usually see it in another cell type in their body. So here we used a cohort where we had both fibroblasts and T cells. And if we called here epivariations in T cells that were losses, the same loci in the same people are usually hypermethylated in fibroblasts. And then the converse was true. So if you had a gain of methylation in T cells, that locus was always nearly very highly methylated in fibroblasts too. And again, very highly significant. So saying basically there was strong concordance here between blood and fibroblasts. We then looked also in another cohort, a small cohort of cadavers where there were multiple tissues available. We called epivariations in the blood, the top left plot in each one. Here you can see outliers, and in the majority of tissues, but not all. So here's one where it's not present here in, I can't read that, I think it's adipose tissue. But usually the epivariations are present to some greater or lesser extent in other tissues of the body. Again, I'm running short on time, so I'm just going to skip these and go to this about tandem repeats. So I started off by telling you the classic example of a secondary epi mutation is the fragile X, and this is caused by this underlying CGG repeat expansion that becomes highly methylated. And so if we just look at, here I'm showing you a table of the top 20 most frequent hypermethylation events we found, just sorted by prevalence in our population. And the last column here says, does this epivariation contain a CGG repeat? And you'll see that the answer is yes for 5 of them. And of 3 of those 5, it's actually already known that these loci have hypermethylated CGG repeat expansions that occur at some frequency in the population. So FRA10AC is fragile site 10AC1. FRA11A is caused by expansions in this gene C11orf80. And this fragile site 12A is caused by CGG expansions in DIP2B. And then you'll notice there are 2 others which have not been previously reported, where we also see common gains of methylation with an overlapping CGG repeat. So then the obvious question is, are these also caused by underlying repeat expansions? So here is this locus LINGO3, where we see a clear gain of methylation in one individual. Here is the CGG expansion, CGG repeat. And we actually had Illumina whole genome sequencing this individual. And you see this very unusual pattern where you see lots of soft clip reads composed of excess copies of CGG. And these red reads here in this IGV plot are unmapped. If you go and look at those reads, they're actually composed of pure CGGs. You compare that to controls where you don't see this. And so this is a very strong signature saying, hey, this CGG tract is much bigger in this person than it is in the reference genome. We then did PacBio long read sequencing, confirmed this individual has about 220 copies of CGG, telling us that this is a novel CGG expansion that's causing this hypermethylation event. Here's this other locus, PCMTD2. We actually observed 38 individuals with this gain of methylation. So that's a pretty common event, 1 per 600 in the population. Again, an underlying CGG repeat, which when we do PacBio sequencing, again, we see reads containing between 600 and 900 copies of CGG. So again, a novel repeat expansion causing this hypermethylation event. Just to note, this is something we consistently see when we're sequencing across these CGG expansions. The PacBio sequencing is chugging away quite happily, hits the expansion, and then the read turns to crap. Probably this is the fact that these are fragile sites with this very strong secondary structure, and the polymerase is having a really hard time when it hits the expanded allele of actually getting through and just can't read the sequence anymore. And then one more, this is FZD6, again, an underlying CCG repeat. This is located at the fragile site 8A locus. Here we did Oxford nanopore sequencing and found the insertion of about 1,800 CGGs, so pretty big expansion. The nice thing about the nanopore sequencing is we could then phase the reads saying, do you have a normal size repeat or an expanded CGG repeat, and show the expansions are specifically where the methylation is. So monoallelic methylation associated with the expansion. And if we just go back, in total, we're predicting probably we found around 30 novel CGG repeat expansions in the genome. We validated 5 of these by either long-read sequencing or southern blot. And there's around 20 other loci we're predicting are probably caused by underlying CGG repeat expansions, which we're working our way through now. And again, just in the interest of time, basically these were explaining nearly all of the unmapped fragile sites in the genome. So with these either validated or novel repeat expansions. I will just skip through this, showing that we prove that basically this gene CSNK1E, which is our predicted repeat expansion, is indeed caused by a repeat expansion found by this other group at the same time. And just to give you the conclusions then from the study, this is really the first large-scale study looking at epivariations in the human genome, and we found these are relatively frequent events, and that a lot of these are doing important things, disrupting the expression patterns of important genes. In particular, we find more than 400 omin genes that have what appear to be potentially inactivating promoter methylation events, including several that are these so-called ACMG genes. And this suggests that epivariations are probably contributing to many different genetic disorders. Our data on mechanisms suggest that around 2/3 of epivariations are probably heritable and caused by underlying sequence variation, and 1/3 are either sporadic, that is primary, or either somatically acquired events with age. We're finding lots of novel, unstable CGG repeat expansions in the genome. I'd just like to emphasize, if you're someone who's doing large-scale sequencing studies, you're not going to be finding these things. You need to be doing epigenome profiling to find them. You may find a random mutation in the genome and wonder what it's doing, but by laying on top epigenetic data like this, you can start to figure out some of these important events. I'll just very quickly say, here's a paper we published 2 years ago where we looked at around 500 trios and characterized epivariations specifically found in those that were absent in controls. Headline number here, we found a nearly 3-fold enrichment for de novo epivariations in patients with congenital disease compared to controls, so saying a lot of these are probably linked with disease. Some of them are really weird and interesting. So here's one. This was actually an inherited event. The father carries the same event as the child. This is a gain of methylation upstream of this gene WDR18. If you knock WDR18 out in zebrafish, it messes up their left-right asymmetry of the body. What's interesting is this particular patient had transposition of the great arteries, that is a left-right asymmetry defect specifically in heart development. We've never seen this in the more than 23,000 individuals we've now looked at. So even though it's inherited, this is the kind of thing that may be linked with their phenotype. And one other thing, We also found many individuals that have, for example, multiple de novo epivariations in their genome. This is one individual that had 11 different de novo epivariations that were absent in parents. We even went back and got a second DNA sample, repeated the array, it's got exactly the same result at all 11, so these are all real. And what we subsequently found is that, interestingly, this patient, when you go back, their exome data then was properly analyzed and found they had a de novo mutation in this gene ADNP, which is part of a chromatin remodeling complex. This all fits together perfectly. Clearly, mutating a chromatin remodeler is messing up their genome in very consistent ways. And it's now known that, in fact, if you have mutations in ADNP, it causes a distinct DNA methylation episignature. I think I saw on your lecture list you had Bekim Sajdakovic talking about exactly this a month or two ago. So we're finding exactly the same kind of things here. And one other paper where we looked at cases and controls in autism schizophrenia. And I will finish there and just say thanks to everybody that helped, and I'll take some questions.
Hi, Andrew. Thanks so much. Very interesting talk. So we have several questions for you. One is for Andrew. Demo. Sorry. Can you unmute yourself? And
yeah, very interesting talk. Wondering about the SNVs that were associated with the epivariation, but were distant. I think you said like 500 kb. Do you have a good explanation? Are they just associated, or are they causing?
Yeah, so what we have there is a statistical association that's, you know, highly significant even after multiple testing. And all it's really highlighting is a haplotype block, right, of, you know, tens of kb. So we don't have a causative variant, just like you get in GWAS, you get a candidate region, but no, you don't know what the causative variant is. Some of these things we're getting hints. So for example, in the study we did in congenital disorder patients, we saw an excess of, for example, mutations disrupting CTCF. And that's known as an important factor that binds to DNA and influences chromatin looping. So when you knock out these CTCF sites, you change the chromatin looping, and which is obviously in many cases going to change the epigenetic. So some of them may be things like this, where they're long range sort of epigenetic regulatory events that they're marking. But bottom line is no, we don't know for sure.
okay. So yeah, that's what's a follow-up question regarding this. So it's really interesting to see that the strong association between the local SNV and also the epivariance. So I was just wondering, you actually did the association study between the epivariation with SNV and also CNV copy number variation. And I just realized that there's only like 2 to 3% CNV actually associated with the epivariations. So if I remember that correctly, and I was just wondering, like, because for my understanding is that there's a lot of epivariants actually associated with the sequence repeats. So I expect maybe there's a higher percentage of CNV instead of like only like 2 to 3%. So my question is that why that happened, and also did you actually have planned to do any other genetic variants, like say structural variation and some other types of variation?
So yeah, probably that's an underestimate. So specifically, we looked at ones relatively close, within 50 kb. It's probably going to capture, you know, a significant fraction of the ones associated, but I'm sure there are other copy number variants located more distally, just like we saw some distally located SNP association signals. We haven't looked at, I mean, we could do all kinds of other studies looking at other types of structural variant, but it gets harder to do, right? I'm sure there are, yes, probably some weird structural variants in inversions, things like that, but they're harder to characterize. So we just focused on the sort of the easy stuff. one thing we're seeing that I didn't talk about here is there's definitely more repeat expansions going on in genomes that I think is underappreciated right now. And I only talked about CGG repeats, but there are other kinds of repeat expansions that are influencing epigenetics. So non-CGG repeats, but also other like VNTRs, things like that, other large repeat structures that are poorly characterized right now. So yeah, I think there's going to be all kinds of events that are underlying these things. Some of them are harder to figure out than others.
Right.
And we probably need other technologies, long read sequencing and things to do that properly.
Got it. Thank you. Yeah, we have several more questions. One is from Nicholas Necomp. Sorry.
Hi. Based on the results you showed, I guess there wouldn't necessarily, or I would guess that there wouldn't be a strong dependence of ethnicity on epivariations. But it also seems like, you know, the cohorts you selected wouldn't necessarily show that one way or another. I guess I was wondering if you identified any interesting secondary epivariations that were over or underrepresented in a particular group.
So yeah, you're right. Bottom line is we don't have very good data on that. I mean, I would just guess, yes, I'm sure there are some that are much more prevalent or unique to certain ethnicities, but yeah, because of the data that we are using, one of the cohorts is mostly Hispanic, and we did see a few things specifically in that cohort that we didn't see in others. But no, we don't really have any good data on that, unfortunately. Most of what we have tend to be Caucasians, just like I say, we didn't generate
what's there, right?
Yeah, but I'm sure there are going to be some that are just like other kinds of variation that are ethnic specific.
Okay.
Yeah,
I have two questions I was hoping to fit in. You showed a little data on the tissue specificity, and maybe hinting that it's going to work to take blood and look at methylation changes in like brain disease or, you know, some kind of other pediatric conditions. I wonder if you can try to extrapolate on that a little bit. And then you talked a lot about XAC, and are we going to have some kind of reference database for methylation variation in the human population?
Yeah, so I mean, we hopefully in the next couple of months, you know, our whole database, I say database, it's really a giant Excel sheet of coordinates, will be published. So yeah, that'll be available for anyone to look at. So your other question was in relation to tissue specificity. So yeah, I mean, there's kind of two schools of thought here. You know, some of our data, or our data is saying that some fraction of these are probably somatically acquired. So as you get older, there's a significant association with people having more epivariations, and monozygous twins, about a third of them are discordant, so saying that's almost certainly somatic in origin. So that would argue that there should be tissue differences. And for some of the epivariations, we do see that, but then for many of the others, we see that it appears to be consistent across tissues. So my interpretation would be the set of epivariations that are secondary, that is caused by an underlying sequence variation, you're going to see that in any tissue in the body. So like imagine Fragile X, every cell has repeat expansion. The methylation mark will equally be there across every cell. Whereas if it's, say, a just purely sporadic epigenetic error that occurs during differentiation or something that, you know, whatever, for some reason these cells didn't methylate that gene properly, that may be only occurring in certain tissues in the body. So we can say with some reasonable certainty what those relative fractions are. It looks like a roughly 1/3 somatic, roughly 2/3 secondary. But for any one epivariation, we actually have very poor data apart from repeat expansions and the small number where we've shown associations. That's only like 5% of the total that we're able to study. So I don't know if that answers your question.
Yes, thank you.
But yeah, overall, it's not a crazy thing to do. Obviously, we can't get brains of people usually. So looking in blood, a lot of the time is going to work. It's not perfect, but it's a pretty decent job, I would say.
Okay, we have one more question from Christy Murphy. Christy,
hi.
Hi.
So I was just wondering how this would work in a clinical setting. So should a physician or a geneticist be testing for these promoter epivariants at the same time they're looking for coding changes, or should they be waiting for the sequencing or del-dup analysis to come back as negative and then moving on to look for these epivariants?
Yeah, so I mean, the testing we did, it's a small fraction of individuals where you could say we found something that we're pretty sure is linked to the disease. So I would say probably, and it's a bit of an art analyzing these data, as I was talking with Anne and Lynn before everybody else came on. Like, this is not like calling SNPs. Like, it's quantitative data. It varies across tissues. Epigenetics varies with ages. You get batch effects. Like, it's not black and white, like saying, do you have a T variant or a C variant in this position? So it's inherently harder to analyze. And yeah, right now, like, our study is really the first study that's even said, where are these in the genome? What are they doing? And who has them? And we still don't have a lot of those answers, right? So I'd say clinically, it's pretty early days. If you want to be really forward thinking and kind of do this in a research context, then you're probably going to find some things in a small fraction of individuals, but you're also going to find a whole lot of stuff that you really can't interpret because they're singleton observations and maybe everything doesn't quite fit perfectly. But yeah, in this study and the two others that I highlighted we've done, yes, they're definitely involved in some fraction of cases. But it's an evolving field.
All right, thank you.
All right. Do we have any more questions from the audience? If not, thank you so much, Dr. Andrew Sharp.
No problem. Thank you. It was a pleasure.
Yeah. Thanks, Anne, for co-host together with me.
Very
Thank you. Yeah, thank you, everyone.
All right, thanks. And yeah, if people have other questions or you're interested in collaborations, feel free to reach out. So I think there's a lot of things we can do resulting from this kind of work. And they'll probably be involved in lots of diseases. So.
All right.
All right.
Great.
Thanks,
Have a nice
everyone.
day. Thank
Cheers.
you. Bye.